This is Neurology Recall, a collection of our most useful, practical, and educational podcasts on a wide variety of topics selected from our neurology podcast collection. Whether you are new to neurology and training or have been practicing for many years, Neurology Recall is designed to help you continue to learn the essentials of neurology, easily, one podcast at a time.
Hi everyone, it's Jeff Ratliff, Deputy Editor of the podcast, welcoming you to the September 2023 Neurology Recall. This month we are featuring a number of interviews we've hosted on the podcast on the topic of amyotrophic lateral sclerosis. These interviews feature ongoing research developments as well as unique patient experiences. The first interview is from the November 14, 2022 episode of the podcast in which Stacy Clardy spoke with Walter Korshetz, Director of the National Institute of Neurologic Disorders and Stroke, on the ALS strategic plan from the NINDS. Next, from the July 5th, 2023 episode of the podcast, you'll hear Gordon Smith discuss simulation-based interim analysis of ALS clinical trials with Jordi van Unnik and Ruben P.A. After that, we'll feature the interview that Matthew Harms did with Emily Plowman on respiratory strength training for patients with ALS that aired on the April 24, 2023 episode of the podcast. Our next interview discusses a newly approved therapy for ALS associated with SOD1 mutations that occurred between Gordon Smith and Suma Babu from the May 18, 2023 episode of the podcast. And finally, we'll wrap up this month's recall on ALS with a conversation, again featuring Gordon Smith, as he spoke with Dr. Ajay Sampat on his experience as a neurologist diagnosed with ALS, which aired in the August 17, 2023 episode of the podcast. And finally, we'll wrap up this month's recall on ALS with a conversation, again featuring Gordon Smith, as he spoke with Dr. Ajay Sampat on his experience as a neurologist diagnosed with ALS, which aired on the August 17, 2023 episode of the podcast. We hope you'll enjoy this recap of interviews across the topic of ALS, and we'll see you next month.
This is Jose Merino, Editor-in-Chief of the Neurology family of journals. The Neurology podcast provides practical information to neurologists and other clinicians to help them provide better care for their patients. Thanks for listening and have a great week.
Hi, this is Stacy Clardy, and today I'm speaking with Walter Korshetz, Director of the National Institute of Neurological Disorders and Stroke. Our podcast listeners will know that Walter has been really quite generous with his time and already gave us specific updates on the state of Alzheimer and other dementias on the April 21st episode this year. And then again, he talked to us on June 20th about other opportunities for researchers with NINDS, including strategies to get involved and get funding, whatever the stage of career, and for all our respective areas of research within neurology. Today, we're going to talk about all things ALS, amyotrophic lateral sclerosis, or Lou Gehrig's disease. Specifically, I'd love to talk with you, Walter, about what is the strategic plan of NINDS for ALS, and what is the state of research into this absolutely devastating neurologic disease. So, Walter, thanks for joining us again. It is so great to hear directly from you.
Well, it's a pleasure, Stacy. And of course, I'm sure all the listeners know how tragic a disease ALS is. The lifespan of people after diagnosis is somewhere around 3 years. I mean, there is variability, but it's a really tragic illness that leads people to lose all of their voluntary muscle strength, eventually unable to breathe without assistance, unable to speak. It is a real tragic disease for the people who get it and for the people who take care of people who have this disease. And NINDS has been working, I guess since its beginning on trying to get better treatments for people with ALS. And as you mentioned, we're reinvigorating things now. We are putting together a strategic plan. And just last week, it's very timely to have the podcast now, because just last week we had our public comment period about the strategic plan. It was put together by a great team of people from all outside the government, from academia, from patient advocacy groups, and we actually had people who have ALS in the strategic planning process, and they went through and developed recommendations for multiple different areas, basic research, clinical trials, how to build collaborations and partnerships, and then research to improve the care of people with ALS. It was a really spirited, I would say, very energetic and very productive series of discussions we had last week. Now, this strategic plan will go out for public comment, and it will be revised based on those comments, and then it comes to our council in February for final approval. But I guess one take-home point is that the term that kept coming up is the ALS clock, that the people who have ALS or people who have genes that predispose them to develop ALS, their clock is ticking, and the urgency is really important that we move as quickly as possible. So we're hoping to actually start things even before, not really even waiting for things to pan out in their usual government manner of approvals. So I think that was really quite an experience. It's on videocast if people want to see it at the NIH videocast. It's really something that I feel really good about.
So for our listeners, we're recording October 31st. This episode is going to air a little bit later, but as you heard Walter say, you have all the way up through February to contribute to the comments. Walter, let's start with some recent news. Again, it's late October 2022, but at the end of September 2022, the U.S. Food and Drug Administration approved a new medication for ALS. It's a combination of two drugs, sodium phenylbutyrate and taurursodiol, and that was shown to reduce the rate of decline on a clinical assessment of daily functioning, and perhaps more importantly, was associated with decreased hospitalization and slightly longer overall survival. So basically, those ALS patients on the drug live 10 months longer than those who were not on the drug. So that brings the total to 6 drugs overall approved for either symptoms or treatment of ALS. What are your thoughts on this most recent approval?
They fit into 2 general groups, I think. First of all, it's just fantastic to have another drug that improves outcome in people with ALS. And it's an oral medication as compared to some of the others that require intravenous infusions. It'll be much more accessible, easier to deliver. In the other bucket, as with the other medications, the effects are moderate. So we have people who have ALS, and what we really want to do is to be able to really slow the progression of disease so that they never get to that final end stage fatal state. And none of these drugs allow that at the current time. So it's really great to have them, but it's not close to what we want for people with ALS in terms of the effect sizes. These drugs were drugs that were tested, beginning in the 1980s and 90s. So they're coming out of research that's 40 years old now. And we really should be able to do better given our knowledge of ALS now.
You hit the nail on the head there, right? It is a few months more, but a few months more for a disease that on average is fatal within 3 years is so little time for the people affected. So what is next from NINDS on ALS? Can you tell us more details about the strategic plan and how you're hoping to make more substantial gains for the condition?
I think there's a couple of things that came out of the strategic planning that we have to wait and see how the priorities come out from the group, but just a couple of interesting things that I heard. One is that there was incredible interest from people who have these genetic mutations but are asymptomatic, knowing that their parents developed ALS and they have the gene, that they're really interested in participating in the research process. Their clock is ticking as well, and currently they feel that they're really not getting a chance to participate. And so it brought to mind some of the work we did in Huntington's years ago. Again, it's a genetic disease, and so there's lots of people who know they have the gene. And we did studies basically to try and understand, given all the factors, when someone is likely to develop the symptoms. And so there were 2 research studies, one called Predict HD and the other was Pharos HD. And the importance of that is that, as you know, in most of the neurodegenerative diseases now, the premium is on treating people as early as possible. And in fact, in the Alzheimer trials now, you're trying to treat people even before they have symptoms. So to do that in ALS or Huntington's, the people with the genetic form give you that opportunity to identify what the factors are that determine when they develop symptoms. And that algorithm would be really important for a neuroprotective trial, because then all you have to do is show you can delay the symptom onset from what is predicted. So that I thought was something that was really interesting that came out of discussions. I think also there's a lot of activity in the ALS field now, so there's more companies interested. There's really interesting basic research we can get to. There's clinical trials going on. There's a platform trial called the Healy platform trial that's testing multiple different agents. So there's a lot going on. I think that the community could benefit from bringing all these things together and kind of coordinating them better and then bring the data together. So a lot of discussion about the inefficiencies of having a whole bunch of silo projects going on that are not coordinated where the data doesn't come in. So I thought that was really helpful. I think in terms of the ultimate goal of getting a really effective therapy for ALS, that really rests on knowing exactly what the therapeutic targets should be that are going to be powerful if you can intervene on them. And so that's going very strongly in the genetic forms of ALS, where you know what the gene is. Then you understand, using iPS cells or animal models, what that abnormal gene is doing to the motor neurons, to the microglia, to the astrocytes. And that gets you targets that you might go after with certain drugs. Also, you have the ability to use genomic therapy to actually do genetic interventions, particularly now the antisense oligonucleotide strategies to reduce the effect of these gain-of-function mutations is going on very strong. And that's something that we have to perfect. It should work. The question is, how early do you have to treat them? And how do you get the ASO where you want it to go? And how do you do that without creating toxicity? So still a lot of issues with that, but the success of the early spinal muscular atrophy trials certainly lights up a lot of hope that this can work. So then the issue turns to the non-genetic forms of ALS, which account for the majority. So probably the highest percentage of genetic is going to somewhere between 10 and 20%, which leaves 80 to 85% as sporadics. And that is really the mystery. What is causing sporadic ALS? So a lot of discussion about that. And it's important because the science field has moved heavily towards the genetics because you can really get your hand around it. You can really do really good experiments. You can see what the therapies are. So it's a much more cleaner package than going after the sporadic. But sporadic ALS is the majority of cases. So how do you get to the bottom of that? Very difficult to know right now. The suspicion is that there are environmental factors that are driving this disease. We have to understand what those are. So there was discussion. There was actually a lecture at the American Neurological Association about environmental drivers of disease. And Eva Feldman talked about ALS and all the potential toxins that could come into play. So getting at the environmental factors, I think, is the holy grail. Difficult, but I think we need to make an effort there. We have to understand, if there are hotspots of ALS, what's the common features in those hotspots, and then maybe looking at populations of sporadic ALS patients to see if there are some common exposures that they share in common. And then the other area, which I think is actually exploding a little bit now, is trying to go from the genetic forms of ALS to the sporadic form. And so the lessons learned from studying the genetic forms, what do they have to tell us about the sporadic forms? And it's true about a lot of our neurodegenerative diseases, Alzheimer's disease, Parkinson's disease, ALS. It's a common theme where the genetics has opened doors for the science, and then the big question is how do those lessons transfer to the sporadic forms? So the thing about ALS is that in most of the genetic forms, not all of them, but most of the genetic forms, there is the aggregation of a protein called TDP-43 in the motor neurons. And that is true about all the sporadic cases too. So the hypothesis is to try and understand from the genetics what is causing this TDP-43 aggregation and what are its untoward effects, with the idea that if you can block those, that will also work not only in the genetic form, but the sporadic form. So that's a hypothesis. There's a lot of assumptions in there, scientific assumptions. Probably have to be lucky for it to work. It is an avenue to explore generically, and I think has, real possibilities for developing inroads to sporadic ALS.
So what I hear you saying in there is maybe the genetics can inform perhaps a bit of the epigenetics, including in their environment and exposures. My next question for you is, you have a challenging position. I'm not the first person to tell you this. You know, all neurologic diseases are tough. They're conditions that we don't like for anyone to have. Certainly they affect over the entire lifespan from birth through elderly. When we talk about ALS, are you able to devote certain resources financially, in terms of personnel, to push ALS a bit further along faster in terms of the research you were just talking about?
You're right. We have 400 different diseases. We can't mount a gigantic program in every one of the 400. So what we do is we look for opportunities in science that will cut across multiple different diseases. So certainly the neurodegenerative diseases at our end, we see a lot of cross science, especially when you get into things like inflammation, which is common to all the disorders, or protein aggregation, which is common to all the neurodegenerative disorders. So we try to invest in the science that a breakthrough there could affect multiple different conditions. And then we look for opportunities where unfortunately, the opportunity may fall into one disease versus the other. So we've certainly seen this with the Alzheimer-related dementias, where Congress appropriated $3 billion additional dollars per year for dementia research. So that gives us tremendous opportunity to do things that we could never do in Lewy body dementia, or the dementia in Parkinson's, and frontotemporal dementia, vascular contribution to dementia. And in fact, ALS science has also derived substantial resources because ALS overlaps with frontotemporal dementia in many people, and that frontotemporal dementia is qualifies for this additional funding that came from Congress. So there, if we get additional funding, then we can do things, I think. We can try and develop public-private partnerships with industry that's interested in a particular area that can help invigorate the funding in a particular area. And then our general way of funding is that we take in grants from the outside and they compete against each other. So I think there's lots of opportunities for ALS researchers. I thought that from what I heard from the strategic planning, we have a mechanism for everything that's been suggested. So what we really need is we need people on the outside to commit, to really work hard on a project that they think is critically important. And we will help them see how to move through our mechanisms. But our door is always open. We take grants in 3 times a year, and we're open to anything that was mentioned in the strategic planning process, for sure. As people may know, in December, the president signed the Act for ALS bill, which is going to have a high impact for ALS research. That bill has 2 main parties in it. One is the FDA and one is NIH, particularly NINDS. So it's definitely working closely with the FDA, which is a great thing. It calls for public-private partnership to be formed between NIH, the FDA, advocacy groups, industry partners, academics. And the FDA has announced that they're going to be working with what's called the CPATH Institute to set this up. I think that's incredibly exciting. The funding for the Act for ALS, some of the money came to NINDS, and we're actually funding this public-private partnership with CPATH and the FDA. The FDA was also instructed to do a grant program, and they've set up a natural history study in ALS that we're also co-funding with the FDA. That's being run out of the University of Minnesota now. And so that's exciting. And the FDA is looking at telemedicine as a way of caring for people with ALS. Very important, given the terrible difficulties with travel for ALS patients into clinics. So all good there. And then also NIH received funding to fund what's called research through expanded access. And expanded access is an approval given by the FDA to make accessible drugs that are still in phase 3 clinical trials. So they haven't been shown to be effective yet. But for people who don't qualify for clinical trials, through expanded access, they can get treated with these medications. And so NINDS had a solicitation for grants, and we're funding a drug called Trehalose, which is in studies at the Healy ALS platform now to treat a certain number of patients with the drug who don't qualify for clinical trials. Who are the people who don't qualify for clinical trials? Generally, the clinical trials want people in the very earliest stages of the disease. So I think the majority of people who don't qualify are people who, finish the trial and so they're done with the trial, or people who are later in the disease process. That project will get started very soon. And so I think developing this public-private partnership, getting more information on, the natural history of ALS, particularly the sporadic form, I think that's going to be very helpful.
Well, there's a lot in what you said there. I'm especially encouraged to hear about the interagency pathways, which are always helpful and accelerating from all different avenues. And a lot of what you said in there, correct me if I'm wrong, but I think a lot of it did come out of that ALS advocacy community as well, right? And listening to what the patients wanted, right? They are well aware of the risks in almost all cases of trying experimental agents, but given the rapidity with which ALS progresses, they really want a shot to try some of these. And it sounds like, the agencies have responded there. So that's good to hear. Along those lines, I have to say I've been really impressed by the advocacy of the ALS community. As you know, I spent a bit of time on social media related to the podcast, and I've been fortunate to follow some of these absolutely incredible ALS patients and their loved ones. Somehow in the midst of their daily struggles, they manage to advocate just so effectively for research and better treatments. And it seems that they are very much being heard based on what you've said. What has been your experience, personal or in your role with the ALS advocates and the advocacy groups?
Well, I think, as a neurologist who took care of people with ALS, as a medical student on neurology, the attending said, if you can take care of an ALS patient, you can become a neurologist. If you can't, you better not become a neurologist. So I think all of us who have seen the suffering due to ALS are so dedicated to try to develop better therapies and help people who have the illness. And I think that knowing what the illness is, one can certainly understand the urgency on the part of people who have ALS. When I was a medical student, the big news was people trying to get snake venom for ALS. So this disease like this is always that hope that something is going to really solve it. But that oftentimes, a false hope. And so people have to be very realistic about what they do, what decisions they make. So for expanded access, for instance, it's uncontrolled. And what we know is that variability in ALS is such that without a control group, you can't tell if a drug is working or not. So we're not going to get any evidence of efficacy out of the expanded access trials unless there's like a gigantic effect size. But if there's a gigantic effect size, they're going to see in the phase 3 trial a lot faster because that's controlled. So it's certainly very understandable that people want access to medications, but really what you want is access to a drug that's going to really make a big difference. And there, one has to really go with the scientific evidence. And so building those targets, those wall of targets for ALS and testing them for high effect sizes, that's really what we need in ALS.
Right, absolutely. I guess to your point, though, in many cases, some of the expanded access participants would not qualify for the trials. Is that what I'm hearing you say?
Correct. No, that's absolutely true, yeah.
Along those same lines, we do have a lot of patients who listen to the podcast, including many ALS patients and their loved ones. Any thoughts specifically for the patients and their caregivers related to research, to the current state of treatments? I know you think about this a lot. What thoughts do you have for that community?
Well, out of strategic planning, what we heard was the people with ALS or the caregivers, they really want to be involved in the research. And like anything that we do here at NIH, we don't actually make things happen. We give grants to people who want to make things happen. So developing that community on the outside that really wants to dedicate themselves to the research effort, that's where I think patients and caregivers have tremendous impact. So getting young neurologists to work on careers in ALS research, for instance, it was brought out of the strategic planning process that we don't have enough neurologists in this area to do the kind of research that the strategic plan is going to be calling for. So we need more people working. We should think about maybe the caregivers. They expressed interest in actually getting into the weeds on these projects and helping with the projects. So that's something to think about. And then, making sure that the research is really as feasible as possible for the patients, which is a problem with ALS and other neurological diseases where the neurologists sit around the table and say, this is what we want to test, and put yourself in the patient's shoes and think about what does this mean for them? Can they actually handle the burden of the research? And that, I think, is probably best informed by people who have the disease or people who have taken care of people with the disease to design the research so it gets to what final endpoint is, but at the least burden for the patients with ALS. And there, I think, telemedicine, I think, would offer a lot as well. I think if we develop wearable devices that chart the progression of the disease, in the home with the patient, with the person who's affected, that could cut down on visits and also help you get to people in rural areas, which is a big problem right now for ALS because the centers are generally, in large metropolitan areas, and people with ALS, particularly in rural areas, they have to drive. Somebody was talking about I have to drive 3 hours to get to San Francisco for my follow-up appointment. And, I could do it when I ended the trial. But now it's 2 years later, I have to pay for almost like an ambulance to drive me to San Francisco. That's just not right. We have to figure out better ways of reducing the burden of people with ALS so they can participate in research.
Absolutely. Yeah, I couldn't agree more. I remember being involved early on in my training in some ALS trials as a sub-investigator, and I was just so impressed, people coming in willing to get infusions and lines placed. And I think, to your point, as anyone who's ever been able to work with the community, just an amazing community, but just because they have done it does not mean we should not try to make it substantially better and easier for participation.
Right. You're right. It's an amazing community. Good point. It really is.
Walter, a lot of information you gave us today. I feel like I've gotten a great update on where the research opportunities are, on where the state of the treatments is, and that is to say there are a couple, but you're very clear they need to get much better as fast as possible. Any final thoughts on ALS, the research, the community, any of it today?
The ALS community is an amazing community, and it is galvanized now to really make a big push in research. I am really gratified by that. I think that NIH and NINDS as the major funder of research, now working with the FDA in a public-private partnership and trying to get industry folks interested. I think there's definitely a level of optimism in a tragic disease that I hope can use and catapult us to finally find something that's an effective treatment to slow down this disease.
Well said, and it's great to hear that coming from you, Walter. Again, we've been talking with Walter Korshetz. He's the director of the National Institute of Neurological Disorders and Stroke. We really appreciate your continued work and leadership for updating us today on the state of ALS treatment and research. And I am so glad to hear that NINDS is appropriately devoting a great amount of effort to finding better treatments. Thank you, Walter.
Well, thank you, Stacy. It was a pleasure talking.
Hello, my name's Gordon Smith. I'm a neuromuscular neurologist and chair of the Department of Neurology at Virginia Commonwealth University in Richmond, Virginia. I'm excited today to have the opportunity to talk to Jordi van Unnik, who is a PhD student at the University Medical Center in Utrecht, and Ruben Van Eyck, who's an assistant professor and medical statistician at UMC Utrecht in Utrecht, Netherlands. Doctors Van Unnik and Van Eyck are going to talk with us today on the Neurology podcast about their article, Development and Evaluation of a Simulation-Based Algorithm to Optimize the Planning of Interim Analyses for Clinical Trials in ALS. It's a long title, but a really exciting article. Jordi and Ruben, thanks so much for joining me on the podcast today.
Thank you very much for the introduction.
This is going to be a really fun discussion, and it's at a really, I think, pivotal time in ALS. Obviously, we've had a couple of new drug approvals, most recently Tofersen, which is exciting. In fact, we did a podcast recently with Dr. Suma Babu from Mass General about this approval, so our listeners might want to check that out after listening to this. Despite this excitement, of course, most clinical trials in ALS and, for that matter, other degenerative disorders have been negative over the years. I wonder if we can begin by telling our listeners what problem you've tried to solve in this study. What were the goals? What were you trying to achieve?
As you highlight already, so the main aim of the study was to think about ways of how can we identify studies that are not going to show any results, so futile clinical trials. How can we identify those upfront or sooner so that you can stop such a study and then hopefully invest the remaining resources that you have and the remaining time that you have into another drug development line. So that's the main aim of the study. And related to that then is what would be the ideal timing. So if you're doing a study, so what would be then the ideal timing to look at whether the drug has shown the intended effect or whether it's already time to conclude the futility.
So, Ruben, there must be a number of benefits to ending a study earlier when it's appropriate to do so because of futility, beyond just saving money and budget. I mean, can you talk a little bit about the other reasons this is so important and objective?
Of course, there's like the time and the money component, but especially for ALS, it's for patients maybe the most important reason. So they want to try out treatment. So if we know already that at one point the treatment is not going to be effective, then the patient has much better chances to switch therapies and try something else out. Another reason is that some drugs, unfortunately, had some safety issues. So what we show in our studies that we actually applied it to 2 trials with severe safety concerns. So there was evidence that there might be a beneficial effect, but in the end, during the trial, it turned out that there was actually a harmful effect of treatment. And I think that that's very important to identify those kind of drug effects very early on and make sure that we have enough evidence and make the right decision to stop such a study. And in that way, prevent doing harm to our patients.
Yeah, Ruben, I really appreciate that answer. I'm just thinking about the context here in the United States. Of course, we have the Healy platform trials going on, which are really exciting to patients because they have potential access to multiple therapeutics. And I don't know, as an administrator, I often tell people, let's fail early and fail often. If you're going to fail, get it out of the way and move on. And I think this is a really exciting approach. So, Ruben, thanks for that great introductory answer. And I wonder, Jordi, if we might just begin our conversation, and we're going to get into more detail momentarily, with what the answer was. What did you find? How did this simulation-based algorithm work? Can you provide a high-level summary for our listeners? And, maybe talk a little bit about your thoughts about how this is going to inform clinical trial design. And I think this really lays the foundation for the rest of our discussion.
What is currently done is that the statistical analysis in most clinical trials for ALS are performed after patients have completed a fixed follow-up. And some clinical trials include interim analysis that are performed after a pre-specified number of patients have completed a follow-up. And in these instances, pre-specified timings together with their decision rules are established to determine when a trial may be stopped early, so either for efficacy or for futility. And these criteria in current trials are often defined arbitrarily and frequently ignore information that is available from earlier trials. So as a result, the trial design may be suboptimal. So what the algorithm does is that it tries to optimize the planning of interim analysis for clinical trials by using re-information from earlier clinical trials. And so by applying this algorithm, we show in our paper that we could reduce the trial duration and the number of patients randomized of all 3 trials that were evaluated by the algorithm. And we believe that the utilization of this algorithm in the design phase of a clinical trial can lead to a better use of time and resources available, but also minimize the exposure of patients to ineffective or harmful drugs.
That's a great explanation. And obviously, it's a complicated approach from a modeling and statistical perspective. I think you really provided some clarity for our listeners who want to learn more. And I hope that's basically all of you. I'd refer you to figure 1 in the paper, which provides a really nice schematic of the simulation-based algorithm that Jordi just walked through and gives you a sense of the impact of modeling the uncertainty of a particular agent. And what I'd like to do is maybe talk about a specific example in a moment. But I wonder if we could step back a little bit and talk more about how interim analyses are currently used in ALS clinical trials. For instance, do all ALS clinical trials currently use an interim analysis or not? And when they are performed, what's the general approach now? What's our current baseline?
So the methodology behind interim analysis that has already been invented, I think, in the 70s or 60s even. So it has been around for very long, and there are many statistical papers that show doing an interim analysis is very effective. So you can shorten your trial duration and save patients being randomized to inferior treatment arms. Despite that this knowledge is well known, only about maybe 5 to 10% of the ALS trials is actually using interim analysis. For me, that's very surprising because the probability that the trial in ALS, or maybe we can generalize it to any neurodegenerative disease, is going to be successful is very low. So yeah, we did once a systematic review, and there we found this low number of trials that are implementing interim analysis. Well, we could have saved a lot of resources, I think, if every trial would implement them. And probably the reason why investigators are a little bit hesitant of using interim analysis is related to their kind of scary statistics. So they require multiple testing, and you need to adjust for that. And they often require specialized software. So there's a little bit of unfamiliarity among clinicians with them. So they're often in the lead of defining the protocol for clinical trials, and what they don't know, they often don't propose, or what they're not familiar with, they don't want to implement for some reason. And I think you often see this especially in the pharmaceutical industry. So the academic trials might be a little bit more liberal. But the other issue is that conducting an interim analysis brings some operational challenges, especially for the statistician, but also for the whole research team, because you need to close a database and you need to make sure that all the data has been input into the database. So you need to take all the charge from the patients, and then research nurse, whoever is inputting the data, needs to complete that into the data set, and then the statistician needs to analyze it. So it's quite some work of conducting an interim analysis. So I think that's one of the reasons for it, especially now with electronic healthcare records, and almost every software package now can generate a interim analysis design for you. So I don't think those borders should limit now trials of using it more, because in the end, using an interim analysis can really improve patient outcomes and also improve the use of resources.
I'm just reflecting on this a bit, Ruben. There's a human element here too, I think, that you talk a little bit in your paper in terms of design interim analysis, or really design of clinical trials. And I think clinical trialists are eternal optimists. We never want to give up on things. And so I've sat on my fair number of data safety monitoring boards and data monitoring boards, and no one likes stopping a trial. And I think you brought a really good reason why we should be more comfortable stopping trials. I think what would be really helpful for our listeners is to talk a little bit about one of the examples. And what I really liked about your article is you developed this methodology, and then you kind of validated it by looking at 3 prior ALS clinical trials, so a couple of diaphragmatic pacing trials and ceftriaxone. And I wonder if we could talk about the ceftriaxone trial and just about how you applied the model. And so if I understand correctly, the way you did this is you looked at ceftriaxone as an agent and thought, okay, this is unlikely to be a home run. And so you inflated uncertainty around the estimates of outcome with the treatment. And then you projected a survival impact, which I was kind of curious about, because you projected what seemed to me a pretty big survival impact. So 28.9 months in the placebo versus 43.4 in the ceftriaxone treatment group. And I wonder if you could just talk a little bit about, at a high level, this example, how you made these sorts of decisions, and kind of what the outcome was for this particular trial and modeling your approach.
So when we design a trial, then usually we have already some kind of preliminary evidence. So often it comes from a smaller phase 2. And this is the case what also was in the ceftriaxone trial. So that there was a seamless design, but we pretended that it had different stages of the development. So what they first did is they conducted a smaller phase 2 trial, I believe in 30 or 40 patients. So they randomized them to placebo and then to the active drug, to ceftriaxone in this case. And then they observe a treatment effect. And then based on the treatment effect, they're going to start to design their phase phase 3 pivotal study. And what we often do is that we just select the treatment effect that we observe in our phase 2, and then we just copy paste that to phase 3, and we assume the treatment effect to be effect. So if we observe there like difference in the survival time, as you indicated, then we just copy that treatment effect onto the phase 3, and then we do the sample size calculations and the duration of the study. But the treatment effect is not given. There is actually a lot of uncertainty around that treatment effect. Else we would not do a phase 3 trial anymore, because else we would already be sure that the treatment would be working. And that's exactly what we did in our study here. Is we tried to see what kind of evidence did the phase 2 trial actually provide. So indeed, it did show a beneficial treatment effect, but how much uncertainty in terms of, for example, a 95% confidence interval was around that observed treatment effect. And then based on that uncertainty, we can then see what is then the probability that that treatment would show an effect in phase 3. And if it's going to show an effect, when would that effect be then? And this is what we try to approach with the simulations. And then on those simulations, then we evaluated, so if this is our uncertainty in the treatment effect, when would the best timing be to conduct an interim analysis? And if you simulate it many times, you can, in a sense, get like the best design or the best planning of your interim analysis to stop the trial as most efficiently as possible. I will give Jordi time to reflect on that as well.
Yeah, thank you. So just to come back to that uncertainty a bit. So what we did is, rather than using a fixed estimate, we defined an uncertainty distribution, like Ruben said, did not make the treatment effect a fixed estimate, but rather follows some sort of distribution based on the evidence that was shown in a phase 2 trial, for example. But we then took it a step further, because we saw that even when the evidence in a phase 2 trial, for example, is really strong, we still see that a lot of trials fail in their phase 3 subsequent trial, so to say. So because of this, we just figured that ALS is a really difficult disease to study, and that it has a really high probability of failing a clinical trial, independent of the evidence that was shown at the phase 2 trial.
Yes, I think that's interesting that you mentioned that indeed, because that's something that happens independent of the ceftriaxone trial. So you have two sources of evidence. So you have the phase 2 that can give you some information about like how certain are you about the treatment effect. But we also know that doing any trial in ALS, there's like a poor probability of success. And that you can just estimate by seeing so how many trials were conducted in ALS in the past and how many of those were successful. So based on that, you can also have some kind of probability of success. So what, like Jordi said, is that we combine those two estimates into like one uncertainty distribution about the treatment effect.
That's a great explanation. I think it really brings into focus what you were trying to achieve and how you went about it. I wonder if we could talk a little bit about timing of clinical effect. And I'm hot off the Tofersen podcast, which was very interesting and a very exciting therapy. And one of the things I found interesting about Tofersen is that during the clinical trial, there was a strong biomarker signal. But the clinical signal didn't become apparent until the open label extension, and those data are available on the FDA website now. How do you integrate thoughts and uncertainty about timing of clinical effect? And it'd be interesting to see how your model would have performed looking at that clinical trial, right? Because there, it may have just been a matter of timing. That's an issue. Is that one of the variables? What are your thoughts about that?
That's a very good example. So what you see in the Tofersen is indeed like what we call like a delayed response. So the separation between the two treatment arms happens after 6 months, that it really becomes apparent. So if you would conduct like a interim analysis after, let's say, 3 months, then indeed it would look like the trial is futile, and then you may falsely stop. So that is indeed the risk of doing interim analysis. But yeah, that risk is in principle not different than from a design where you don't do any interim analysis. So that risk is balanced there. But I think that is really a realistic scenario also for ALS, because usually it takes time, and we can see it in Tofersen, but we can also see it in other drugs for ALS, or Riluzole, for example. It just takes some time for a biological response to translate into a clinical response. It's unlikely that if we give a treatment today in an ALS patient, that tomorrow we can already show like a slowing of the disease. Probably it's going to take, well, indeed, like 3 or 4 or 5 months. So that's something we didn't really incorporate into our design, but it can be quite easily extended just by saying, well, we define like a minimum time duration that we want to see follow-up of the patient before we're going to start doing interim analysis. So I think that's the easiest way to implement it. But also on the technical side, I think that we can modify the simulation models was saying, well, we know like the uncertainty around the treatment effect. We know the probability of success in ALS trials, but we also know from few trials how the treatment response would appear if there is a treatment response. So that third level of uncertainty into the simulations might also be another strategy.
It seems to me, Ruben, that you use not only estimates of certainty or probability of a treatment effect, but data from preclinical and early clinical development, knowledge about the pharmacology and the target of the drug. And I think this is going to evolve over time, isn't it? The more we learn from hopefully more successful trials, and you'll be better able to design the right algorithm.
Exactly. So now we picked only one outcome. In our study, it was that we looked only at survival. But yeah, if at one point, so in case of Tofersen, there was like a very strong biomarker response. So if the biomarker becomes a very important outcome, then maybe we can also do like early interim analysis based on the biomarker. And if we don't have enough evidence for futility or efficacy at that time point, then we switch to the clinical outcome that you make it even more adaptive, the strategy.
You brought up the term adaptive, and one of the things I found really interesting in the discussion on your paper is how this approach might be used for other purposes. So you talked about, for instance, modification of eligibility criteria. And ALS is a highly heterogeneous disease, as implied in our earlier conversations and as our listeners know. And I wonder if you could talk about other ways this type of approach to interim analyses might be useful. And it seems to me, this seems tailor-made to integrate with adaptive trial design or Bayesian trial design. I wonder if that's the case, and maybe you can blue sky how clinical trialists can use this approach in innovative ways.
Yeah, so we agree that this is only the beginning. During our simulations, we use the interim analysis only to see if the trial could be stopped or not. But like you said, we can use the results of interim analysis to do many things. For example, to enrich the trial by changing or specify, at least more restrictive with the inclusion criteria to only include the patients that we see an effect on.
So I think that especially for ALS, that latter part is very important. So we see that probably genetic subgroups are responding better to certain therapies. So we see now the evidence of Tofersen, but there's also other trials in ALS that might show a different response among certain gene carriers of ALS genes. So another adaptation during an interim analysis could be to just decide, okay, I only continue with, let's say, the C9orf carriers in ALS, and then I drop all the other patients. So that I think that kind of adaptations is what you meant as well, right?
Yeah, I did.
Well, guys, thank you so much. And I just want to encourage our listeners to check out this article. It's a really interesting read. And I think Jordi and Ruben, you've done a great job of not only describing the approach in a way that's understandable, but the implications of this. And as I've pondered it, I think there are really great implications, large implications, not only for ALS clinical trials, but how we can apply this across therapeutic development. And I think what's really exciting about it is not just stopping for futility, but in a world where we have an explosion in therapeutic development, there's a real premium on deciding when you're going to stop a trial, because there are lots of options for our patients who really want to be part of discovery and have access to novel therapeutics. So congratulations. Truly appreciate you taking the time to talk and make this understandable. And I'll encourage our listeners to check out the article. It was just published in the journal Neurology. So Jordi and Ruben, thank you so much.
Thank you for having us.
Thank you very much for the invitation.
Hello, everyone, and welcome to the Neurology Podcast. I'm Matthew Harms, a practicing ALS neurologist at Columbia University, and it's my privilege today to be able to focus on an exciting paper coming from the ALS literature that's now published in Neurology, titled Respiratory Strength Training in ALS, a double-blind, randomized, multi-sham controlled trial. Here to talk with me today is Emily Plowman from the University of Florida, where her team really is leading the way and focusing on both bulbar and respiratory function in ALS. As many of you know, if you've seen ALS patients, it is often the respiratory decline and the bulbar function that ends up being the major determinant of survival. And so these are really especially important points for clinical care in this patient population. So it's a real pleasure to have you here today, Emily.
Thanks for having me, Matthew.
Your paper just came out, and focuses on using respiratory strength training in ALS. And so I was hoping you might share the main points and methodologies of your paper.
Firstly, just to backtrack to let you know, what motivated our work in this area. We've been, from a broadly speaking standpoint, traditional sort of ALS models of care have been primarily palliative and reactive in nature, whereby interventions have been applied after and in response to clinically identified impairment thresholds. So, for example, as you know, the use of mechanical insufflation, exsufflation, or cough assist has been routinely prescribed in the United States once a critical impairment threshold whereby peak cough flow hits 270 liters per minute. And so reactionary way, then all of a sudden this cough assist device is prescribed to the patient. And this historical reactive model of care is really disease-centric. And what I mean by that is that the unknown and sometimes unpredictable ALS disease progression, in addition to a clinician's ability to identify those impairments, which, as you all know, sometimes access to care can be an issue, particularly with the recent COVID pandemic. And so it really leaves the patient and the family sort of helpless and at the mercy of this sort of unpredictable, ravaging disease. And we've been thinking in our group here at the University of Florida for some time that we know that there's unfortunately the eminent development of dyspnea, dysphagia, and dystassia or cough impairment. And so the historical model, reactionary model, doesn't take advantage of potentially the early ability to improve physiologic capacity and functional reserve for vital functions such as breathing, airway clearance, and oral intake. And so our line of work in this area is largely born out of a desire to examine if a proactive approach that flips this model a bit on its head, and rather than the disease dictating models of care, the patient is educated as best we can with what we currently know. And then it empowers them to make a decision early in the disease. But this is specifically for early affected individuals with ALS.
I'm thinking about the fact that when patients come to see me and they have limb weakness, very quickly say, a toned, fit muscle will decline less quickly. So definitely organize your life so that you're keeping muscles that are strong, fit, and toned. I guess I don't have the tools or haven't thought about ways to make that happen for bulbar or respiratory muscles. And is that what you're getting at with this training?
As we all know, use it or lose it. And we're reminded of it when we go to the gym and it takes us a while to build up nice muscle tone and strength. But the minute you stop doing that, unfortunately, it tends to go away. And so the broad approach here is to actively engage muscles and strength and physiologic reserve, specifically that do target cough and airway clearance capacities, and then also breathing and hoping to stave off respiratory insufficiency, which, as we know, is the leading cause of death in individuals with ALS.
So start off by walking us through how you picked your intervention, what it looked like, kind of what an average participant would experience doing these exercises and trainings that your study focused on.
We screened 51 individuals with ALS across 2 sites at the University of South Florida in Tampa and the University of Florida in Gainesville. And we enrolled 45 early affected individuals with ALS. And I would like to specify that these patients, the inclusion criteria, they had to have a forced vital capacity that was greater than or equal to 70% predicted. So that's a very important point to make out when we're talking about our findings, that these don't generalize to every individual with ALS. In addition, their ALS functional rating scale revised needed to be 34 or higher. And we specifically targeted this group, Matthew, because we do speculate that there's this critical window of time in which engaging in these proactive exercises could be beneficial to pump up the bank account, so to speak, or the savings account, which is sometimes how I describe it to patients, which unfortunately they will decline, but we're just sort of trying to boost up their physiologic capacities, similar to what folks may do before undergoing a surgery. They may be told that they need to physically get fitter or perhaps lose weight to give themselves the best post-operative outcomes. But in this way, we're increasing their physiologic reserve and bumping up that curve, of which they'll then unfortunately decline, but we're very interested to see, given the nature of the terminal illness and the importance of breathing, airway clearance, and swallowing for survival, if this could have a functional impact. And really the first question was, is it feasible? Is it safe to do a respiratory strength training program? And then secondly, can we increase maximum inspiratory and maximum expiratory pressures, which are the latter being very important for cough and airway clearance, and the former being, we would think, very important for staving off respiratory insufficiency.
Tell us what the training looked like, what it consisted of, and then tell us what you found in the results.
The training specifically, as I mentioned earlier, we call it RST, or respiratory strength training, and our protocol involves both expiratory and inspiratory sets of exercises. And typically historical programs have just focused on one or the other. For this, individuals were enrolled in our study, they underwent baseline testing, and then they were randomized, to either go into the active respiratory strength training group or the sham training group. It's really important for this to be practical for the patients. So they did the therapies at home, and we sent a home therapist out 1 day a week. In the past, when we had our pals come into clinics, we would get a lot of drops because, it's just too much of a burden on them. And so that's an important component. They would complete their exercises 5 days a week, and it consisted of using 2 different trainers, one, like I mentioned, for the expiratory muscles and the other for the inspiratory muscles. And these are one-way spring-loaded pressure threshold devices where a patient either blows into the trainer for the expiratory repetitions, or they breathe in through the trainer for the inspiratory repetitions, and they have to successfully complete a repetition. They actually have to blow through this pressure threshold device. We can, much like going to the gym and doing weight-bearing exercises, we can actually load the inspiratory and expiratory muscles with these one-way calibrated spring-loaded devices. So we specifically, for this trial, loaded the muscles at a 30% individualized load for the expiratory and inspiratory systems. It's sort of important to point out here that historical load-bearing exercise programs that follow sports medicine principles will progressively load the muscles, and typically starting around 60% of their 1 rep max, moving up to 70 or perhaps even 80%. Given that this is a progressive and terminal disease that includes muscle wasting, one very important point is that we don't want to overtax or overload the muscles. So they did daily 25 repetitions of their expiratory repetitions and 25 repetitions of their inspiratory repetitions. So folks did this for a 12-week period. They had weekly home therapy visits whereby a therapist would retest their inspiratory and expiratory maximum pressures and recalibrate the trainers so that they were always at that 30% load. And then following their completed 12-week exercise program, they attended the laboratory here at UF and completed their post-RST assessments. And then we also looked at 1-year functional outcomes for time to referral to non-invasive ventilation, looking at both their bulbar subscale and total ALS decline rates over the 12-month period, and then also their oral feeding status.
And if I understand from your paper correctly, you found that doing this training was associated with improvements in some metrics and no change in other metrics. Can you walk us through that aspect of the results?
We specifically noticed for our short-term outcomes and our primary outcomes that the program led to an immediate physiologic improvement in maximum expiratory pressures, as well as cough function. However, it didn't affect maximum inspiratory pressures or forced vital capacity. When we looked at our functional 1-year outcome data, we noted that the ALS FRSR bulbar subscale decline rates were attenuated for individuals in the active RST group, and specifically those in the sham group, their slope or monthly average decline rate was almost 3 times higher or faster than those in the active group. I would like to point out, because, when we publish these papers, there's a lot of heavy reliance on statistical significance. But what we found to be highly relevant and meaningful to the patients in this trial was that although at the beginning of this study, all patients were able to eat orally by mouth, some form of intake, when we looked at our 1-year, 12-month outcome data, 87% of individuals in the active group were still consuming an oral diet compared to only 64% of individuals in the sham group. And so we found that to be clinically quite meaningful to those patients. And then the other thing I'd like to point out that potentially could be missed by the statistical significance, but although our survival analysis didn't denote any significant differences for timing of NIV referrals across group, if you looked at data on the 14 participants who did commence non-invasive ventilation, on average, those in the active group commenced NIV 12 months later than those in the sham group, suggesting that perhaps respiratory function was preserved for a bit longer in these participants.
Do you have another trial in mind? And how would you do things differently based on what you learned from this?
I think, the key is we need to increase and do this across other sites. Like a lot of studies, this in some senses raises more questions than answers. And so something that we noted here in this particular trial was we believe there could be a bit of a dose response in terms of the loading that we're putting on the muscles. Further work needs to look at, well, what is the actual ideal load that we would want to place on the muscles to maybe induce or optimize physiologic gains in function in early affected patients. We really need to figure out, what are the key sort of ingredients to optimize this in which patients and at what dose.
I, for one, would love to see these types of studies funded more. I'm sure you and your lab would also.
Oh, yeah, absolutely.
I do have the sense, though, that these types of interventions, although not as attractive to funding agencies as antisense oligonucleotides or interventions that are, molecularly driven, are really important and are getting some traction.
This is very low tech. It's very easy to disseminate because the trainers that one can use for this are typically in the order of like $50 for patients that they can even order them themselves. They can be trained. We've recently done studies using telehealth as well with, the necessity to do this during the pandemic. But I do think it's starting to get traction. And I'll be honest with you, I do agree. Like I've always felt these more disease hard-hitting basic molecular studies would get more traction. But I recently had the fortune of being, as I know you were too, Matt, on the NINDS ALS Strategic Task Force to promote, future directions for funding. And I was part of the quality of life panel. And I'll be honest, I sort of thought, no, people are not going to be, everyone's going to go take their lunch break during our session and there's going to be less interest in that. But I was pleasantly surprised. I found the opposite. And I had a lot of people contact me afterwards. And so I do agree with you. And I'm excited about this, that right now people are living with these diseases and, what pals and their caregivers have, but they've taught me a lot. But what they also have shown me is, the human spirit sort of needs a sense of purpose every day. And, they also need a sense of hope, and not to overstate this because this is sort of symptom modifying in the short term. So I want to be clear on that. But I have noted in hundreds of patients that I've done this with, even just in clinic, that when they know they have to get up every day and do their exercises and they have a purpose with that, there's also like a intangible sort of quality of life aspect to this, I think, that is really nice. And the fact that it empowers them to take control of one thing. And they typically see improvements in their numbers. And when everything else is frankly sort of, going down the tube, then this is like a really great thing for them. So, yeah.
Well, fantastic. And really appreciate you taking the time today to talk to us about your paper, Respiratory Strength Training in ALS, a double-blind, randomized, multi-center, sham-controlled trial. Obviously a mouthful of a title, but need to fit all of that in there to convey what it does. That paper with Emily Plowman as the first author is now available in pre-publication in the journal Neurology. Thank you again for joining us today for the Neurology Podcast. We hope you check out other episodes on the webpage.
Hello, my name's Gordon Smith. I'm a neuromuscular neurologist and chair of the Department of Neurology at Virginia Commonwealth University. And I am excited to have the opportunity to talk today with Suma Babu. Suma is an assistant professor of neurology at Harvard Medical School and MGH. She's a physician investigator interested in ALS. She's specifically interested in SOD1 related ALS, hereditary ALS, which we're going to talk about today. Dr. Babu is going to talk with us about the recent FDA approval of Tofersen for ALS in the setting of an SOD1 mutation. So this is a super exciting topic. Suma, thanks so much for joining me today. I'm really looking forward to our conversation on the podcast.
Thank you so much for having me.
Let's cut to the chase, right? This is super exciting. So what is Tofersen? The brand name is Qalsody. But in any case, why is it such a big deal in the ALS community, and how is this going to change your practice?
This is a very, very timely and important question. And Qalsody is the first antisense oligonucleotide or gene-targeted therapy that has been FDA approved for a certain subtype of ALS caused by a genetic mutation due to SOD1 mutation. And this is very, very exciting for all of the ALS community. SOD1 mutation itself contributes to about 2% of all cases of ALS. And there are some subtypes of SOD1 ALS that are extremely aggressive. And this is autosomal dominant inherited gain of function mutation. And I've cared for several families where family members have passed away within 10 to 12 months from symptom onset, consistently, generations after generations. And with the approval of this gene-targeted therapy, there is so much joy in the community and hope for a potentially life-saving therapy for these families. And secondly, beyond SOD1 ALS, I think Tofersen or Qalsody really brings hope and is paving the way for other forms of ALS that could also benefit from other similar gene-targeted therapy approach. And in fact, there is also an antisense oligonucleotide trial in phase 1 that has already started for all forms of ALS or sporadic ALS outside of US, and we're waiting to start that in the US as well.
Before we get into what Tofersen is and the evidence, I wonder if you could just personalize this for our listeners. I've been doing neuromuscular medicine for longer than I care to admit, and there's been at times a sense of nihilism. What's different this time? Why is Tofersen different from other therapies we've seen?
What I've seen, several of our participants in the trial, as well as the expanded access protocol that followed after the trial, have complete halt in progression of their clinical disease. There are a handful of participants at our site that I've seen who their neurological examination today is exactly the same as what it was when they first entered the trial, either in 2018 or 2019. 2019 was when the phase 3 trial started. And so I've never seen anything this remarkable in any of my ALS patients otherwise. And these are individuals who carry a very fast progressing variant of SOD1. And these are individuals who have had family members, as I mentioned earlier, pass away within 10 to 12 months historically. And this has been a game changer for these families, and they're actually changing the history for their families. It's been a very, very rewarding experience. And I think that's part of the excitement that comes with this approval. Sometimes we go beyond the statistical significance and p-values that we see in clinical trials. And when we see remarkable benefit in more than a handful of patients firsthand, I think it brings so much hope and promise for the ALS community.
Well, that's super exciting. There's been a history of getting excited about small numbers of patients who have stabilization. And in fact, your group has published on this in large trial data sets. But this certainly sounds like a very different experience than what has been seen in earlier clinical trials. So that's very exciting. Maybe before we get into clinical trial and the FDA approval, can you tell us just briefly how this agent works?
Tofersen or Qalsody is an antisense oligonucleotide that is specific for SOD1 type of ALS individuals. And what this ASO does is it binds to the toxic mRNAs, and it then allows for these toxic mRNAs to be degraded via the RNase H pathway and cleaves the toxic mRNAs and prevents the synthesis of the toxic SOD1 protein. The toxic SOD1 protein is the hallmark, and it's really at the core of what causes the problem in SOD1 ALS. And by preventing it at its grassroots level, so to speak, we're actually modifying the disease biology and rescuing motor neurons and reducing neurodegeneration. SOD1 is a vital gene for cellular function. So the aim here is not to knock out the SOD1 gene completely, but actually it is to reduce the production of the toxic form of SOD1 protein only. And that's what Qalsody does.
So let's pivot and talk about the FDA approval, which took the accelerated approval pathway, which allowed Biogen to get approval based on a surrogate endpoint measure, and in this case, the neurofilament light chain levels. And I wonder if you could talk a little bit about neurofilament light chain, because that's a hot topic across all of neurology, across all neurodegeneration and central and peripheral nervous system, but really important here. What's the evidence that neurofilament light chain is a good predictive marker of clinical response in ALS?
This is an evolving topic, and I think as a community, we're all learning still about how to use neurofilament levels in various different domains of clinical practice as well as research. And our understanding is deepening with every new study that is being published about this, and the utility is also evolving. Now, to put things in perspective, Tofersen trial in the VALOR trial, the VALOR study was a phase 3 trial of Tofersen that actually was then submitted to the FDA for approval. It was a 28-week double-blind study. And following the double-blind study, there has been an open-label extension that has been continuing indefinitely. So the predetermined outcomes in the 28-week trial included a pre-specified neurofilament light chain level as a marker of neurodegeneration. And this trial showed that in people who were treated with Tofersen in the trial experienced a 55% reduction in plasma neurofilament light chains, compared to the placebo group, where a 12% increase was noted over that 28-week period. And this was a very nominal p-value of less than 0.0001. And that was then submitted to the FDA under the accelerated approval pathway, because at the 28-week mark, the trial did not meet its pre-specified primary clinical endpoint of ALS functional rating scale change. Now, there's more complexity to this, which I'll get into in a little bit, because the clinical change appeared much later, and that was not noted until the open label extension. This is one of those drugs that actually takes a long time to show clinical benefit, and the biomarker benefit preceded the clinical benefit. But in either case, neurofilament was a marker that led to the accelerated approval. FDA has this unique provision for accelerated approval, especially for serious or life-threatening diseases, if there is a surrogate endpoint that is likely to predict clinical benefit. And taking into account the severity, the rarity, and the prevalence of the condition, and what are the available alternative therapies at the time, taking all of this into account. So the accelerated approval pathway is an option that the sponsor of Tofersen decided to pursue to get this FDA approved in an expeditious manner. Now, what we know about neurofilament levels in ALS is that neurofilament light chain is not specific for ALS alone. It is a marker of neurodegeneration. It's a protein that leaks into the interstitial fluid and then into the blood, the spinal fluid, once there is a motor axon loss. And this is seen in multiple neurological conditions. But what we know in ALS is that the levels remain fairly stable once phenoconversion has occurred. And there is a wonderful familial ALS cohort that was followed by Michael Benatar at University of Miami for several years. And he showed in his research work very beautifully that these neurofilament levels remain at low levels, normal levels, until the point of phenoconversion. And it turns out that neurofilament levels actually rise even before clinical symptoms appear. And once the clinical symptoms appear, the level stabilizes. And depending on whether an individual's disease has a fast or slow progressing course, it may stabilize at different levels. So fast progressors tend to have much higher levels of neurofilament compared to slow progressors. And compared to other neurodegenerative diseases, there are papers that have shown that neurofilament levels actually exceed in ALS compared to nearly all other neurodegenerative diseases. So until the Tofersen trial, neurofilament level was used as a biomarker in research to determine phenoconversion in SOD1 or other asymptomatic gene carriers. But Tofersen trial is the first one that actually showed that neurofilament levels can actually be lowered by a large percentage. 55% is a large percentage of decrease following treatment effect, following a disease-modifying therapy. So now the field is evolving towards whether we can use neurofilament light chain as a marker of treatment effect. And there is evolving understanding about the utility of neurofilament light chains as a prognostic marker as well in ALS, whether this could actually predict a change in clinical function even before the clinical function occurs. But one has to be careful about how we use neurofilament light because, one, it is not specific for ALS, and two, there is on an individual level some variability in terms of change over time as well as the baseline levels for individual A versus individual B. So on a group level, on a large group level, very clear that it is a clear marker that can be used for phenoconversion, and now with the Tofersen as a marker of treatment effect. But on an individual level for routine clinical practices, I think we have to be a little careful. There are recently other therapies in ALS, for example, Relivrio showed a clinical benefit but did not actually move the neurofilament level as such. So I think this is where our understanding about neurofilament is complex and it is evolving. I think we'll need more evidence to solidify this.
So it sounds like the timing and context are going to be really important. Timing in terms of disease progression and understanding the importance of NFL levels and their change. Could you tell us more about the open label extension? The New England Journal article didn't show a very impressive clinical outcome, but you mentioned that data coming out of the open label extension study suggested a more significant treatment effect. Can you tell us about that?
The most recent data was presented at the March 2023 FDA Advisory Committee meeting, and all of that is available on the FDA website as well. So just to take a step back and talk about the open label extension design itself. Open label extension trial is for trial completers who have completed the 28 weeks of the double-blind portion. And these are participants who continued on from both the phase 1 multiple ascending dose trial as well as from the phase 3 Valor trial. And this is a trial that has been ongoing almost indefinitely since 2017 when the first open label extension was initiated. And in the Valor trial particularly, there were a total of 95 participants who entered the open label extension. And out of these 95 participants, 63 participants, that is 88%, actually received Tofersen during the initial double-blind portion. And 32 participants received placebo. So the folks who actually received Tofersen initially were called as the early start group, and folks who rolled over from the placebo group into the open label extension were called as the delayed start group. And what we know is that at the 52-week mark, and this is using the more recent data cut that was presented at the FDA AdCom just before the approval, the primary endpoint was the ALS-FRS-R or ALS Functional Rating Scale change. And there were clear separation between the two groups in the early treated versus the delayed start group at that week 52 mark. And there was a p-value of 0.02 that was achieved with a difference of about 3.5 point change over that 52-week mark in ALS Functional Rating Scale. Now, in addition to the ALS Functional Rating Scale itself, there were multiple secondary endpoints that favored early start Tofersen over the delayed start Tofersen in the open label extension. For example, the respiratory function, which was measured using predicted slow vital capacity, also favored a slower decline in early start Tofersen group at that week 58 and showed a statistically significant difference of p of 0.01 at that week 52. Secondly, including the quantitative muscle strength testing using handheld dynamometry that also showed a statistically significant group difference in early start versus delayed start group at that week 52 mark with a p-value of 0.01.
So what about patients with sporadic ALS? I think there are probably, 400 patients in the country who have an SOD1 mutation in ALS, but many, many more who have sporadic ALS. You must be getting a ton of questions from your sporadic ALS patients about the Tofersen approval. What are you telling them about this?
This is one of those situations where there is a lot of joy in that very small subset of ALS patient population with SOD1. But what does it mean for the rest of the ALS community? What does it mean for 98% of the ALS community beyond SOD1? We are getting a lot of questions about this. And, what is really exciting right now, where we are in the ALS space right now, is that there are more antisense oligonucleotide or gene-targeted treatment trials that are emerging beyond SOD1 itself. ASOs are sort of like lock and key mechanism, and it works only for a specific indication. So currently there is a combined phase 1-3 treatment trial for a different genetic subtype called as the FUS mutation, fusion and sarcoma mutation. This is also another very aggressive form of ALS, and this is the only form that actually can even affect adolescents. Most other forms affect adults only. So it's exciting that we're able to offer a gene-targeted treatment trial for individuals with FUS mutation. There is another one that targets ataxin-2 repeat expansion, and the one that targets ataxin-2 repeat expansion is designed in such a way that it also is open for sporadic ALS patients because of some preclinical data that suggests that reducing ataxin-2 protein could have a modulating effect on TDP-43, which is almost universal in ALS individuals except for the SOD1 gene mutation positive individuals. And then thirdly, there is another ASO treatment trial that has begun for sporadic ALS, particularly from a company called Curalis. This is a molecule called QRL201, and that treatment trial is in phase 1 and has already begun its first cohort enrollment in countries outside of US, and we're waiting for the US FDA to give permission to start this trial here in the US as well. And even beyond Tofersen and ASOs, this is a number that keeps changing continually. And the last time I checked, there are over 160 companies that are developing therapies for ALS.
Well, Suma, thanks so much for a truly remarkable podcast. What an amazing time this is in the field of ALS and neurodegenerative disease and neurology in general.
Thank you so much.
I want to thank our listeners for listening to today's podcast and a very exciting episode. For those who are interested, you can find the Tofersen phase 3 clinical trial in the New England Journal of Medicine. The first author was Tim Miller. And you can find the open label extension data, which I found particularly exciting, on the Food and Drug Administration website. And again, thanks so much for joining us on a podcast. Of the podcasts I've done, I found this particularly exciting because it really captures the energy and what's new in neurology and why it's such an exciting time to be caring for patients with ALS and other related disease. Hello, my name's Gordon Smith. I'm a neuromuscular neurologist, and I'm chair of the Department of Neurology at Virginia Commonwealth University. And I'm really excited to welcome Ajay Sampat with us today on the Neurology podcast. This will be a really special podcast, I think. Dr. Sampat is an assistant clinical professor of neurology at UC Davis. Ajay has kindly joined us today not to talk about sleep medicine, but to share with us his journey as a patient with ALS. He was diagnosed with ALS last year at the age of 36, I believe. And so, Ajay, thank you so much for joining us. I think this is going to be a really great conversation.
Great. Thank you, Gordon.
I wonder if we can just begin by having you tell us a bit about yourself. I think part of this story is really going to be who you are. Tell us about your family, your day job, and so forth.
Before this diagnosis, I was like any other healthy young person. I had a perfect life, really. I have a wife who's also a physician. She's a retina specialist here in the Bay Area. And then we have 2 kids. So a daughter who's 3 and a son who's 1. And as you can imagine, we had a very busy life with our young family, but it was a really perfect life. Nothing going wrong when ALS entered our lives. And then professionally, I'm a sleep medicine clinician and really a clinical educator. And education is really something that I truly valued in my professional life. And I really had a wonderful position at UC Davis.
Thanks, Ajay. Maybe we can get right into it. I mean, no one, when they're in their 30s, early in their career, young family, is spending time thinking about their mortality or diagnoses like ALS. So what was that like? Maybe you can take us through your diagnostic journey.
Yeah, I mean, I think my diagnostic journey, Gordon, was probably atypical compared to most individuals with this. And that's because of my professional background. So most patients, I believe, take about a year to get diagnosed from their initial symptom to final diagnosis. For me, it took about a few months. And I think that expedited workup was probably a reflection of my medical background. So my symptoms started with cramping actually in my right hand, cramping that was by itself, not terribly worrisome, but then that progressed then to later having fasciculations or twitching. And then later on after that, having actually a weakness in my hands. And so, that prompted a whole cascade of workup that patients with ALS go through, right? The blood work, the urine work, the imaging studies, the genetic studies, the CSF studies, all that stuff. And it took me about 2 months to get a final diagnosis when everything else came back normal except for the EMG.
What was that like? And I know that's a broad question, and there are multiple different components to it, but I'm curious what was it like for the couple of months of uncertainty? And then I'm particularly curious about how the diagnosis was shared with you, what that was like as a neurologist, as someone who's given this diagnosis to more people than I care to count. I think there's a lot to learn from hearing your perspective about that.
It was an overwhelming period of time, as you can imagine. I mean, the stages of grief that we all learn about, in med school or at some point in training, right? The denial, the anger, the negotiation, the depression, acceptance, all of that is very, very real. And I never experienced it until I went through this myself. For me, denial was a relatively short phase, I think, compared to other people, again, partly because of my medical background. But all the other components of it was very real when I was going through this workup. It was the negotiation, if I have this disease instead of that disease, I would be fine. Angry that I was even going through this in the first place. And then depressed that I was watching my colleagues and my friends go about their normal lives while I was getting this workup done. All of that came at the same time at some points, but happened in a very real way. For me, getting the actual diagnosis, I think it was probably March or April when I completed the workup last year and got the official, diagnosis. And at that point, I had seen my workup. The writing was kind of on the wall, and it wasn't necessarily a big shocker, as I think it is for other patients who go through this. And my neurologist was fantastic. I mean, she was very thorough. She was very thoughtful. When she went over it with me, she was doing it with the knowledge that I had probably researched some of this on my own, but she wasn't making assumptions. And I think that was really helpful. I'm not a neuromuscular specialist. I'm a neurologist, but I'm not an ALS expert. And when she was delivering the diagnosis to me and counseling me through it, it was nice to not have those assumptions be made, even though I'm a physician with a neurology background.
So I'm curious when the diagnosis was raised with you. My experience is a lot of neurologists are reticent to raise ALS when they first think about it. And I've even seen people who refuse to tell patients until they refer them into an ALS clinic. Did she bring it up with you earlier in the course?
Yeah, I think it was fairly early in the diagnostic workup. And again, being a neurologist, I kind of knew what was going on and what we were ruling out and what studies we were doing and why we were doing them. So it wasn't a secret. Early on, she brought it up with me and we had a very candid back and forth discussion about the differential that we were ruling out. I don't know honestly how she would do it with other patients or how other doctors do it with non-neurology patients. But I think I really appreciated the honesty, the candor. I mean, she told me as we were ruling out other conditions like MMN and different genetic things, SMA, she told me the possibility of these things are low, but we can still do the appropriate studies to make sure it's not that. And I appreciated that. It was very an honest approach.
Any other take-home points for that or tips or pitfalls to be mindful of, not only in giving a diagnosis of ALS, but many of our diagnoses, even those with exciting therapeutics, are still frightening diagnoses, Parkinson's disease and whatnot. Sounds like compassion, transparency, not making assumptions, other wisdom.
Gordon, I've been giving these talks to residency groups, around the country now about my journey and experience kind of like this. And one big theme I talk about with the residents and medical students is empathy, empathy versus sympathy, and why empathy is really critical to being a good, compassionate doctor. What I would say, my sort of general takeaway as a patient is really take a few seconds or a few minutes and just pause and try to put yourself in the person's shoes. And that's really what the heart of what empathy is. Try to put yourself in their shoes. If you were potentially given this diagnosis, if you potentially in a month could not work, If you potentially in 3 months couldn't dress yourself, if you potentially in 6 months couldn't drive your car, or in 12 months, couldn't actually stand up anymore, how would you feel? Take a few minutes just to think about that reality. And that's what the patient in front of you is going through. And I think if you can do that and pause and reflect, I think that empathy comes to the surface. And that's what I try to tell students and residents when I give this talk. And that's probably the number one, advice, or I guess pearl I would try to convey.
And reflecting on that, I wonder if maybe you can talk about your interactions with colleagues and others, right? It's hard for many people to know, how do I engage with a colleague who has a difficult diagnosis? One of my dear friends was Ted Burns, who's the father of the Neurology podcast, who we lost to cancer last year. And I talked with Ted about this. I think maybe this gets to the sympathy, empathy. How have you navigated professional relationships? And again, tips for what you as a patient who's going through this journey appreciates in your professional relationships?
Yeah, I've been blessed in many ways, despite being very unlucky with this diagnosis. And one of those ways is my department and really the School of Medicine has been very supportive and encouraging. I approached my chair fairly early on and talked to her about what was going on, and approached some of our leadership, and they were very supportive about reassigning clinical weeks and things like that. With my other colleagues, quite frankly, Gordon, I haven't been around, I haven't been going to the clinic, and I don't see a lot of people anymore. And so I've lost a lot of those connections. But there's been a few colleagues that have stayed in touch with me, mainly the ones who are involved in the educational part of the department. And I have honestly really appreciated their just checking in with me, just emailing, texting, how are you doing? Not about work stuff, but just how are things going, right? And that's actually been really nice and really great to still feel like I'm part of the department, even though I'm clinically not doing much, right? But still feel like I'm an active member and engaged. And so what I would say, to anyone that has a professional colleague or even a personal friend or someone that goes through significant medical hardship like this, I would say, just stay connected. Just stay connected with them. I think when I was first divulging my diagnosis, I had an outpouring of support, dozens of calls and emails. Everyone wanted to see how I'm doing, but it all fizzled out. After a month, everyone got back to their busy lives, except for the few that stayed in touch. And those few are the ones that I really I look forward to talking with them because they've stayed in contact with me.
Ajay, as we were talking ahead of our conversation, you mentioned that you continued to see patients for a little while after your diagnosis. What was that like? And did your perspectives on patient care change having gone through this as a patient?
I saw patients probably for about a month or 2 months while I was getting the workup done. So after my official diagnosis, actually stopped seeing patients after that. But for a few months, I was seeing patients while, this workup was being done. And my approach to patient care, I think, has changed in the sense, if I were seeing patients again, I think I would really consider those humanistic principles that we all learn about in first year of med school. It's one of those core doctoring courses that we all take. Listening to patients, communicating with patients, empathy, these big things that a lot of us, frankly, we go through residency training, fellowship training, busy attending life, and we lose sight of a lot of it. For me as a patient, all that stuff from first year medical school came back to the surface. How to talk to patients, how to listen to patients, how to hear their story. And that, I think, is what I have remembered and how I would change my clinical practice if I were still seeing patients.
Tell us about your mentorship and training of medical students now.
I'm one of the academic coaches at our School of Medicine, and what that means is I'm given a cohort of students each year, and I mentor them from first year to fourth year. I told my students fairly early on, too, what I was going through, was very transparent and open with them. And, it was a mix of reactions. Some people very much calling me and texting me, and other students really didn't know how to respond to that, which I get. It's a very serious thing. But over time, over the last year and a half to 2 years, I think I have really cultivated this very special relationship with them. And we've talked about that, that of course I'm there as their faculty mentor to help them with residency applications and things like that. But a lot of them have said, hearing you go through this illness has shaped my impression of what it's like to actually care for people. And for me, that's incredibly rewarding. As a faculty mentor, advisor, I feel like I've gotten this very special relationship with them as part of my role in the department still.
When I think of you personally and having read up about you before talking, one of my first reactions was, Ajay, that you're courageous. This takes courage, right? The advocacy, but to go in front of a bunch of medical students, mentor them and have that conversation. And I wonder whether that's the right response or the wrong response. I think we think of our patients as being courageous, but I don't know. Does that make you feel good or bad? Hear me say that.
Well, a lot of people have told me that, you're so brave and strong for doing this. And I mean, I can see where that's coming from because it's not easy, I think, to talk about difficult topics like this. But for me, Gordon, I give lectures. I like giving lectures, right? It's something that I've always enjoyed doing, actually. One thing that I talk about in my talks is when patients face a very serious illness like this, I think it's important as doctors, as neurologists, to help them find a renewed sense of purpose. And I gave a talk to the UCSF palliative care group a month ago, and a lot of them commented on the same thing, that when we have our patients, most of whom are oncology patients, that's a big thing that we try to do for them. This is my renewed sense of purpose. I can't do the clinical things professionally that I love to do and that I used to do. But for me, this gives me some sense of purpose. So yes, it is courageous, I guess, in one way, but it also helps me combine an interest, a strength of mine, with something that keeps giving me some fulfillment and meaning.
So Ajay, I wonder if we could pivot a little bit and go from maybe purpose to identity. I often think of patients who have chronic and significant medical illness. It becomes part of their identity. Yes. How has this changed your sense of self-identity?
Oh, it's completely destroyed and recreated a different sense of identity. A disease like ALS changes you completely. And like I said, before this, I was a healthy young person, young father, active. That has completely changed with a disease that is physically debilitating like ALS. So my role in my family has changed, right? And I was a young father actively doing things. And with ALS, I've been put on the sidelines. So my identity as a father has changed tremendously, not being an active participant in the sport of parenting, but really being a sideline person on the bench. My identity as a doctor has changed, right? I'm still able to teach and do lectures and things like that, but I'm not able to go to the bedside and show students what hyperreflexia looks like in a bedside patient, or not able to show a patient in the clinic how to read their CPAP report. I'm not able to teach in that way. So my identity has changed as a doctor. And again, I think part of it is figuring out how to recreate an identity that gives you purpose and meaning. And I've tried to do that with my family, right? I can still do some things. I can still read bedtime stories. And so I've sort of tried to focus on the things that I can do still instead of the stuff that I've lost. And same with my professional life. But it has changed tremendously.
We just did a podcast on the Tofersen approval, which is super exciting. And there's been some therapeutic development in ALS. Obviously, some of the therapies are not as effective as we might like. Tofersen's really exciting. What's your perspective on all of that? And I always think there's been tremendous advances in our understanding of mechanism and genetics, but tremendous frustration that that's not translating into therapeutics. And you're a neurologist. I know you focus to sleep, but you're still a neurologist and obviously understand this space pretty well. As a neurologist who has ALS, what's your view of what's going on in the field right now?
I think it's never a good time to have ALS, but I think if you're going to have it at any point in history, now is the best time to have it. As you mentioned, Gordon, there have been a lot of exciting drug development recently. 3 drugs have come out in the last 1.5, 2 years, right? We have oral edaravone that came out last year. We have the Relivrio that came out last year, and then Tofersen that was approved this year. And so we only have 4 FDA-approved drugs for ALS, and 2 of them really have come out in the last year, right? IV edaravone was 2017. But it's been very exciting in that sense. And we have another drug on the docket for an FDA advisory committee meeting next month. So there's a lot of potential excitement. And I think Tofersen actually was a very exciting development because it's been one of the only things so far that has produced upstream effects, targeting genetic causes of this. And I think the actual effects in the trials and patient anecdotes have been very convincing. So I think it's a hopeful time. It's an exciting time. I think once we learn more about the genetics and can make more personalized therapeutics like ASOs, antisense oligonucleotides, that might be more helpful. But I think it's still a hopeful time for all of us.
Ajay, for our listeners who haven't heard it yet, I do want to refer them to a really exciting podcast with Suma Babu, who's an assistant professor at Harvard MGH, about Tofersen. It's a great podcast. And it was exciting to me because I went into that, Ajay, only knowing the published data, but there's additional data, as you know, that's available through the FDA, and that's a really exciting approval. If I may, I want to ask maybe a hard question or maybe a bit of a somber question. You're facing your own mortality now, and I'm wondering how you're going about that, right? I mean, you've made choices professionally that it's difficult for you to see patients, but you're able to teach and you're passionate about that. How is that impacting your life more broadly? You did a really interesting podcast for those of you who want to hear more for ALS TDI, and there you talked about your bucket list. I actually really like your bucket list, and I think our listeners would too. Maybe you can share that.
Because I was lucky in that I had an expedited workup, I had a whole year last year where I could focus on these personally important things like my bucket list. One of which was to visit all 30 baseball stadiums, or to finish visiting all 30 baseball stadiums, which I did last summer. I also had a bucket list item of going to Antarctica, which I was able to do last December with my wife. And we took many, many trips last year, many trips, Albuquerque, Hot Air Balloon Festival, went to Disneyland with my daughter. We went different weekend trips here and there. And that was very important to me, to be able to travel when it was a little bit logistically easier to do so. And so to answer your first part of that question, Gordon, facing my own mortality and how that's been, I mean, for me, again, that denial stage of grief was not a long period because of my background, I believe, or maybe my personality, I don't know. But when I sort of confronted this diagnosis, and of course, as a neurologist, I know how this is heading, right? I don't know the time I have. I don't know the progression, sequence I'm going to have. But knowing this is a very serious terminal condition, I was intentional about how I spent my time, which is why I stopped clinically working. I directly wanted to spend time with my family, to do projects, legacy projects that were meaningful to me. So I've written handwritten letters to my kids. I've written a book for them, done video messages for them, things that, for me was helpful to spend time in a way that was meaningful. And I think anyone facing their own mortality, right? Or if you have a family member or anyone facing a serious condition like this, I would just ask them the simple question, what is important to you? You have a patient in your clinic, you're not sure what to do, just ask them what's important to you. And that's what you should be helping them figure out, whether it's their family, their work, something else, all of the above. Ask them what's important to them. And that's how you should be spending your time.
So Ajay, life is a terminal illness, right? We all face our own mortality. It's all a matter of probability.
Yes.
I wonder, what would you tell your 34-year-old self or 30-year-old self about how to approach priorities? And I guess what I'm really getting at is, what would you tell the 56 or 57-year-old Gordon Smith about how to manage my priorities, right? I mean, what lessons have you learned looking backwards, and how those of us who don't yet know what's going to take our life or when it's going to take our life, how we might approach kind of our prioritization, our own bucket list?
Well, I would say, like with ALS, they say make a bucket list and do the physically more demanding things first and save the least physically demanding things for later. And I would say that's a general good rule of thumb for anyone thinking about their ultimate bucket list or mortality, right? Do the things that you may not be able to do 5 years from now. And most people have a sense of their own physical limits and abilities. Spend the time on doing that now. And of course, I think it's helpful to make a list for some people, but just figure out what you can't do later and do that now. And, there's a quote, I think many people have said this, but I mean, the biggest mistake we make in life is thinking we have time, right? We all do this, whether you're 30 or 60 or 80, people think they have time. And the fact of the matter is, like you said, Gordon, life is a terminal condition. We have less time than we think. And do the things that are important to you now that you won't get to do. And a prime example of that, Gordon, I'll tell you, is on my trip to Antarctica. I obviously couldn't do a lot of physically active things, but there were older individuals on that trip in their 70s, 80s, right? They weren't able to do a lot of things. They enjoyed it like I did, but they weren't able to go kayaking. They weren't able to go on shore. Do that trip in your 50s if you want to do it. Do it in your 60s. Do it when you physically have the ability to do it. Don't think you have time to do it later, because you may not.
Well, Ajay, I very much appreciate you spending time with us today on the podcast, and best of wishes in your journey. And I know our listeners wish you the best as well.
Thank you, Gordon. Thank you for your time as well. And thank you for the invitation to come speak with you.
This is Stacey Clardy, your podcast editor. If you've enjoyed the podcast, please take a few moments to subscribe, rate, and review the Neurology Podcast through Apple Podcasts, Google Podcasts, Spotify, or wherever you listen. And remember, you can always head to neurology.org/podcast for our full list of past episodes. Or you can also search by keyword in your podcast app for any neurology-specific topics you want to learn about.